So this is the last part of chapter number 7. And our glycoconjugates are an example of a glycoconjugate is what's known as a glycoprotein. A glycoprotein is a protein with a small oligosaccharide attached to it. Now, this carbohydrate is attached via its anomeric carbon to an amino acid on the protein. Now, we haven't talked about amino acids at this point on the course. We will get to them, and so we'll revisit this a little bit. But the common connections occur at serine, threonine, or asparagine residues. So these are 3 amino acids of the 20 amino acids that are proteinogenic amino acids. About half of mammalian proteins are glycoproteins. Now, only some bacteria glycosylate a few of their proteins. Carbohydrates play a role in protein-protein recognition of one another. Now, viral proteins are heavily glycosylated, and ultimately that's what helps them evade the immune system. So an example of what this could look like. So what I'd like you to recognize is this right here and this right here. This is our protein. These are our proteins, or a portion of our protein, where we have this group and this group that is found within the protein sequence. Now, what happens is this whole molecule right here gets added to one of the amino acids in this protein sequence. This whole group right here gets added to that amino acid within the protein sequence. Now, what happens with those is we've got this single glucose derivative that's been added. But what do we know about those glucose derivatives is that they can form more complicated molecules. And so we've got this key right here that has a total of 5 different glucose derivatives that can be used to build upon one another. So again, here is our serine or threonine residue within the protein. And to that, you can have a very simple modification. This one shows a galactose molecule and a glucose derivative. So you've got 2 sugars that have been added to the serine or threonine residue. So it's a fairly simple disaccharide that's been added. On the contrary, you can have this complicated network that is spun off of that one serine or threonine residue. Or you can have all of these other different variations of, or all of these other polysaccharides that are added to single amino acids within a protein sequence. Now, another example of a glycoconjugate is the class of molecules known as glycolipids. Now, lipids are one of the other major molecules that we will talk about this semester. And they are, they're a major part of membranes, and lipids can be covalently bound to oligosaccharides. So they are parts of plant and animal cell membranes. In vertebrates, ganglioside carbohydrate composition determines your blood group. So this glycolipid, or it is a glycolipid that dictates what type of, or what blood type that you have. Now, in gram-negative bacteria, LPS, or lipopolysaccharides, can cover the peptidoglycan layer. So whenever you talk about gram-negative versus gram-positive bacteria, you're talking about glycolipids. Now, what will that look like? Here, what you've got is this complex lipid structure, and a modification to that gives you, well, this fairly complicated sugar that stems off of it. Now, proteoglycans are another example of glycoconjugates, and they are sulfated glucosaminoglycans attached to large rod-shaped bacteria in a cell membrane. Examples, syndicans, glipicans, these are proteins that have a single transmembrane domain, or a protein that is anchored to the lipid membrane. Now, these are going to interact with a variety of receptors from neighboring cells, and they're going to regulate cell growth. So these glucosaminoglycans are ways in which cells communicate with one another to be involved in the cell cycle, be involved in growth, the size of the cells specifically, or basically it's glucosamine or proteoglycans, which are used for cells to communicate with one another. So as an example, here is a syndecan, and here is a glypican. Now, a syndecan has a transmembrane domain. We'll talk about this more when we talk about protein sequence and protein structure, but this is a part of the protein that is embedded within the transmembrane domain. Okay, here's the N termini of the protein, here's the C termini of the protein. Again, we'll revisit this later on. But this protein's length has all these modifications coming off of it. And all of these modifications are sugars. On the contrary, a glypican does not have a portion of its protein sequence that is embedded within the membrane. Instead, it has a modification that is a lipid anchor that inserts it into the membrane. And so we'll talk about this again whenever we talk about lipids a little bit later on. But those are two ways in which a proteoglycan can be associated with a membrane. Now, this glypican has all these modifications stemming off of it that enable it to communicate with adjacent cells. So I think that this really well illustrates the complexity of biological systems. For a very long time, there was this idea of, well, if you can understand something about protein structure, awesome, then you're going to know everything. But then if you think about, well, what about these sugars that are attached to those proteins? Are they going to influence the protein structure? They are. Okay, if you have one glucose molecule versus one N-acetylglucosamine, or two N-acetylglucosamines, is that going to influence the protein's function? Absolutely. It's going to impact the contacts that cell or that protein can have with other cells, with other proteins. So at every step in the process, the more that we know, the more that we realize we don't know. So this, I think, illustrates the tip of the iceberg that we're on with respect to sugars and proteins and everything like that. But nevertheless, we have this kind of models with which we can talk about how cells interact with one another. We're going to be revisiting these later on whenever we discuss protein structure and membrane-associated proteins. But until then, this is the end of Chapter 7.